Recall when we talked about meiosis, we learned that chromosomes line up along the middle of the cell during homologous chromosomes during metaphase 1, and they line up independently or randomly. So we don't always have like all of grandma's chromosomes on one side and grandpa's on the other, or in this case all the red on one side or all the blue on one side. There are random arrangements when sperm and egg are made during meiosis. And this is called independent assortment, the principle of independent assortment. And the implication for Mendel's work is that he observed that you don't always have to have dominant traits inherited with dominant or recessive with recessive, that you can have a tall pea plant, which is a dominant characteristic, that has white flowers, which is a recessive characteristic. So we can have an arrangement that looks like this, or we could have an arrangement that has all the red on one side. There are many, many different possibilities, and especially when you think of us that we have, as humans, 23 pairs of chromosomes, there are many, many possibilities of the way that these chromosomes can line up and arrange themselves during meiosis. And that's just one way that we end up having lots of genetic diversity in our population, because of what we have in our sperm and egg. So far, up until this point, we've only looked at the inheritance of one trait at a time, but in our body we have 30,000 traits. So we don't just inherit one trait at a time. So we're going to make it a little bit more complicated and talk about what's called a dihybrid cross, looking at two traits at a time that are inherited. Di meaning two, hybrid meaning a cross between something. If you think of a hybrid car, it's a cross between an electric and a gas car. Dihybrid cross is looking at the inheritance of two traits at a time from two parents. So far, everything we've looked at so far has been a monohybrid cross, looking at one trait from two parents. So you have to be very careful when you're looking at these problems and read them carefully to see what it's asking before you start going and writing things down. So in this particular question, it's asking you, it's saying in pea plants, round seeds are dominant over wrinkled seeds, and yellow seeds are dominant over green seeds. And they want you to determine what you would expect when you have two pea plants, each of them are heterozygous for both traits. Each of them are heterozygous for seed shape, and each of them are heterozygous for seed color. So I'm just, before I even get into what's listed down here, when I'm looking at this problem, I want to kind of figure out what it's asking. Heterozygous for seed shape. So if it's heterozygous, it's going to be capital R, lowercase r, heterozygous for seed shape, round, and heterozygous for seed color. So the dominant seed color is yellow, so capital Y, and it's heterozygous, so I'm going to use lowercase y. And both parents have that genotype. Be careful, because sometimes the problem's going to give you parents with two different genotypes. In this case, both of them are going to be heterozygous at both traits. So heterozygous for round seeds and heterozygous for yellow seeds. And in this problem, they're just giving you the space here to write what we just did. So I'm going to write capital R, lowercase r, because it's heterozygous for seed shape, and I'm going to write capital Y, lowercase y, because it's heterozygous for seed color. And again, in this case, both of the parents have the same genotype, but be careful, because that always won't be the case. So capital R, lowercase r, capital Y, lowercase y. Those are the genotypes of my parents. Now remember, when sperm and egg are going to be made, that these are going to segregate or separate from each other. The capital R and the lowercase r are no longer going to be together like they are in the rest of the cells. When you make the sperm cells, these are going to separate from each other. And that was Mendel's law of segregation. So when these separate from each other, we're going to make gametes that have both of these traits in them. And I find the easiest way to set this up, it takes some time to do, but I use, you might be familiar with this technique from your math classes, but FOIL. And what FOIL stands for is first, outer, inner, and last. So I take this genotype here and I look at my first terms, my first R and my first Y, and I put them together. So that's my F of my FOIL, capital R, capital Y. And then I take my outer terms, my capital R and my lowercase y. And then my inner is my lowercase r, capital Y. And then the last are my lowercase r, lowercase y. And that's how I get what my possible gametes are. And I'm just going to transfer these here because they have the place for them here. So again, my first terms, capital R, capital Y. My outer terms, capital R, lowercase y. My inner terms, lowercase r, capital Y. And my last terms, which is the lowercase r, lowercase y. And then I do the same thing for the other parent, and as you notice, this is going to be the same for this parent, but again, it doesn't hurt us to do this again. I'll use my FOIL technique again. First, I have my capital R, capital Y. I have my outer, which is my capital R, lowercase y. I have my inner, which is my lowercase r, capital Y, and then I have my last, which is my lowercase r, lowercase y.